everyone. I'm Swapnil, and I'm going to talk about B-cell epitope prediction and antibody and PCR structure modeling related part of the talk. So here's the outline of my talk. I'm going to talk about, going to go through a quick introduction on B-cell epitopes. We've been talking about epitopes since yesterday, so I don't need to dive into that too much. I'm going to talk about the schema, how these tools are arranged in the IEDB website. But again, it's pretty similar to what you have seen for MHC binding tools, so that also that will go quickly as well. Then I will dive into the sequence-based epitope prediction methods that we have for linear epitope prediction, and then the structure-based, which are for confirmation or discontinuous epitope prediction. And after that, we'll take a break and then come back for computational antibody design, where I'll focus mainly on antibody and PCR structure modeling, and then give you some hints on antibody design. We'll go through a quick flowchart on that. So as we've been talking about MHC binding tools, the B-cell epitopes are, well, the antigens directly are recognized by B-cells, so there is no processing step in here, no presentation, but antigen in its native form actually binds to B-cells. B-cell receptor, which is the antibody. In this case, this is a glutamine HA1 chain. You can see that only the HA1 chain interacts with the antibody and not the HA2 chain. So this is, well, an important piece of information. Because the epitope is on the HA1 chain, the epitope is actually in direct contact with the antibody. That part of the antibody is also known as paratope. But then what's interesting about this epitope is that it's discontinuous. So sequentially, if you look at it, it's dispersed. The epitope residues are dispersed, but just by the folding process, in this protein, these epitope residues have come together to form this binding site. But it's not the only type of epitopes that are there in the antigens which are recognized by B-cell receptors. There are, of course, linear epitopes as well, but it is estimated that 90% of the B-cell epitopes are discontinuous. But one good thing about the sequential or continuous epitopes is that even after the maturation, you can actually get an antibody response to these. So as I said, I will quickly walk through the schema of B-cell epitope prediction tools. So you have seen this homepage now multiple times since yesterday. You can access the B-cell tools by going down this drop-down menu, selecting B-cell epitope prediction, or clicking these links directly. So let's say I'll go from this link. You'll come to this page. All the linear epitope prediction tools are linked to this. Epitope and ELIPRO are the discontinuous epitope prediction methods. And then we have a simple flowchart with some tools mentioned for the antibody design. And then this is the tool, LIRA, which is for antibody and PCR structure modeling. So again, I'll go through this quickly because you have seen it too many times now. So if you click on this link here for linear epitope prediction, you'll come to this home page. You'll see, again, pretty similar things, input fields where you can put the Swiss prot ID or actually enter the sequence. You have the methods listed in here, TEP2, and then other amino acid scales that Bjorn mentioned in his talk in the morning. Of course, if you want to know more about these methods, you can go to help, or want to see the format that you can enter, like the sequences or IDs you can enter, you can go to the help page, get information on the amino acid scales, actual values in there, the different score thresholds that we use, all the information is here. Of course, you can go, if you want to see a result page quickly, you can go through these examples, ready examples. You just submit the job on the page, and then you'll see the results. Of course, there are citations listed in the reference tab. You can course, download some of these tools. Not all of them are available, unfortunately, but some of them, well, most of them are available to download and use it for standalone purposes. Again, industry, people from industry might have to get a license for these, but for academic use, it's complete. These tools are completely free. And of course, again, we've been hammering this since yesterday. You're always welcome to contact us, and we'll get back to you with answers to your questions if we could as soon as possible. With that, I'll move on to the sequence-based epitope prediction methods. Before diving into that, again, I guess Sinu and Sandeep mentioned these points, but actually when you would want to use epitope prediction methods, is that when you have actually searched through IEDB and you don't see any information available on epitopes in the protein that you're interested in, then of course, by all means, you can go ahead and use epitope prediction methods. But then there is a second scenario as well. Even though the epitopes are available in the IEDB, you might want to know all antigenic determinants, all epitopes present, could be present in the antigen of your interest. Then of course, that's also a good reason to use prediction methods. But of course, you should first go through IEDB, see what information is available. If that's not good enough, go for prediction methods. And there is a reason for this, particularly for B-cell epitopes, that I'll come in the conclusion, come back to it in the conclusion part. So as Bjorn mentioned, the linear epitope prediction tools we have, for historical purposes only, I guess, we have these amino acid based tools, scales there basically. So why we have these? Because some of the features, these were correlated with the presence of continuous epitopes in the proteins or antigens, and that's why features such as beta turn, surface accessibility, flexibility, antigenic propensities basically count occurrence of residues in epitope compared to the other surface residues, and hydrophilicity. So there are different scales which were used, I should say. As I'm pointing out, these only provide you with the information on what might be of interest to the antibody for binding to the antigen, but these are not by standard definition prediction tools. These are not optimized to predict epitopes. So there is, of course, a second class of prediction methods that we have. So Epipred, the 2 version, and there are other methods available in other resources, other websites that you can use to predict linear epitopes in B cells. B-cell epitopes. These machine learning methods actually take polynomial training data sets. You apply, get some features, including these amino acid scales, input those to machine learning methods, as Bjorn and Sandeep had already talked about this, and then predict epitope and optimize prediction on training data set, tested on a completely blind testing data set. So with that, I'll move on to how you can use these tools. Now, of course, if there are any questions, please feel free to ask in between as well. So I'll go through an example here. This is again the first example. If you click on the example tab, take the first example. I guess that's what it is. So this is Epipred is the default method that we recommend. There is a new version of Epipred 2 now, but this is still in testing, so we are still removing some bugs in it. But it should be up and running in a week or two, hopefully. But till then, I would recommend you to use Epipred. So Epipred has been trained on 2 amino acid scales, which are Parker's hydrophilicity and Levitt's secondary structure, and then it uses SMM model to actually to get scores for overlapping peptides, and then assigns these scores to the middle residue of that peptide. The reported AUC is 0.66. And again, as Bjorn mentioned, this is like for MHC binding tools, you have seen 0.8, 0.9 AUC values for B-cell epitope prediction methods. This is the range 0.6, 0.7, somewhere around there. So if you submit your sequence, or actually if you have a Swiss Pro ID, you can enter it here, choose the method of your choice, submit the job, and you'll come to the results page. Now there are two things which you can change: window size and threshold. I would not recommend to change window size, but if for score threshold, it's pretty important thing. If you want how many candidates you want to predict, based on that you can change these thresholds. This threshold value 0.35 has been tested for sensitivity of 0.49 and then specificity of 0.75. So if you want more specific or less specific results, you can adjust your score thresholds. This table is provided in the help tab, so you can always go back to it, see, okay, how many candidate epitopes you are getting. If there are too many, you can always increase the threshold, get less number of candidates for testing.
Can you say specificity? What do you mean by that?
So specificity basically means you are not getting false positives. So let's say this epitope, which has been predicted as an epitope, so this part of the protein which has been predicted as an epitope with candidate epitope, I should say, with this particular score threshold might be wrong, right? It's a prediction after all. So this is known as a false positive, and then that's it. Like you'll have 0.25 or 25% of the false positives. So it's like reducing the number of false positives that you get and include it. So there has to be a balance between these two things. Sensitivity is basically synonymous with accuracy, and this is usually what you would want is higher specificity, but then you lose sensitivity along the way. So that's why the recommended method, recommended score threshold is somewhere in the between, where you have balance of both. But you can.
Just looking at the result, it doesn't make much sense, because you have the result, you have predicted peptides, and the number one is just one.
Yeah.
Then
So
you have, I mean, the program is not working properly, I would say, because you have first a window size of 7, and then you have results of a single amino acid. That doesn't make sense.
So these scores are calculated based on a window of 7 amino acids. So this score that you see just for one amino acid is based on still flanking amino acids combined, scores combined. But then in a way you're right too. Like if you're actually going to validate it experimentally, or you want to do mutagenesis, you won't consider these residues. But it's just a part of like a threshold, right?
This is that an obvious improvement will be to remove from the results, one single amino acid. That's what it is.
Yes, true.
True. Because which of those that are not
well, again, just to reiterate, because these results are based on a score threshold, we don't want to exclude anything. To show, okay,
but you should. That's my point.
Yes. If you, well, as I said, if you want more specific results, you can always increase the score threshold and get less. Of course, by doing that, you'll get less amount of candidate epitopes. If you scroll down the page, you'll see two tables, one for the predicted epitopes, and then for each position in this protein, their scores, and then the assignment. If the score is above the set threshold, it will be assigned as an E, which is for a candidate epitope. And if you scroll down further on the page, you'll get a link to download these results into a CSV file. As I mentioned, there are of course other amino acid scale methods in there, such as Chow Fastman, Peraton, prediction. For these amino acid scale methods, the threshold by default is an average score of a protein that you have entered. So that's usually the cutoff. And again, you can adjust this cutoff based on the requirements. Like if you see too many epitopes, reduce it. Oh, sorry, increase it. The other format is pretty similar, the table format. Of course, you can use multiple amino acid scales in there. But if you are, by any chance, going to use amino acid scales instead of recommended Epitope method, which is actually a prediction method, I would recommend to use a consensus of different methods rather than relying on one, which might work a little better. But I would recommend to use Epitope for predicting linear epitopes. So there has been a study from our group in 2007. So this study actually evaluated all these amino acid scales and showed that none of these amino acid scale methods gave an AUC above 0.6. So let me remind you, 0.5 is a completely random prediction. 0.6 is close enough, it's not very good. And this is attributed to the benchmark data sets, and this has been issued with the B-cell epitope prediction, whether it's a linear or discontinuous epitope prediction. Because of the benchmark data set, in the benchmark data sets, you might have these long peptides where actually only few of the residues are actually epitope residues which directly contact the antibody. But the other residues are also taken in the training data set, and that might actually reduce your accuracy. So if you have any questions on the linear epitope prediction tools?
For the other several methods, like on your slide, you are given the predicted residual scores. So it will only give you the residual scores instead of the predicted peptides. Like for the first method.
Oh, yeah. So yes, it will give you like a position in this, and then the rest, and then their scores. But this is based on the window size again. So this is a score for this peptide. You can assign it to the middle residue. So with that, I'm going to move on to the structure-based prediction, discontinuous epitope prediction methods. Before going to this section, I would just like to point out that so these are the three main methods to actually determine the 3D structure of a protein. If you have an antibody-antigen complex and you would want to determine the structure of it, you would do either crystallography, NMR, or electron microscopy. And most of this data, well, almost all of this data is deposited in PDB, which is the primary data base for 3D structures. But we have 3D antibody-antigen complexes already curated, and you have much more information such as CDRs, the particular epitope residues. We have calculated epitope residues based on 4 angstrom length cutoffs in these exports file that were mentioned by Randy yesterday in the complete database exports, and we have 1791 3D PCR assays in there. If you want a little bit more information on these complexes than what PDB provides, you can go and access this CSV file quickly. So this is the link to the PDB. I just want to mention how to query this quickly if you are interested in getting some information out of it. So this is the home page, and there is a search button here. You can either do a keyword search here. There is a search button here as well. So let's see, you just go here, click on sequences. You can actually enter your sequence here. It will do a blast search. Of course, you can go to advanced sequence search. You can adjust the parameters of the blast if you would like. to get the structures for the sequence that you have entered. And let's say you have narrowed down to this, you have provided a sequence of this, you got this entry. Now, if you want to download a sequence file of this structure, you will go download files and then download the FASTA sequence. And then there is a PDB format, which provides atomic coordinates of these structures here, so you can download that. And this can be given as an input to discontinuous epitope prediction tools. There are other formats, MMSIF, which is now maintained by PDB. So if you scroll down the same page, you'll get information on the chains and what molecule or particular protein it corresponds to. So this is an important information. Sometimes you might need it. What if the results, if you are searching for a particular sequence in the PDB, you don't get any results? Then there is a way to actually model these protein structures or predict these protein structures, but I'm going to talk about this a little later. So coming back to the discontinuous epitope prediction methods now. These methods rely on geometrical properties combined with some other features such as amino acid scales. Geometric properties are, of course, extracted from 3D structure of a protein. So methods like Discotope, Eliprove, one of the first method, which was CEP. And then there are other methods which use different training machine learning approaches, PSVR. And then some of the protein docking algorithms were also used for B-cell epitope prediction. And I'll give you an overall idea of these, but I'm not going to go into details about these tools. And of course, there are some sequence-based approach which predict discontinuous epitopes, but again, I'm not going to cover this as well. So Discotope is one of the tools that we recommend at IEDB for discontinuous epitope prediction. We have two versions, Discotope 1 and 2. So Discotope 2 was actually trained on 75 crystal structures of antigen complexes, and this particular version actually took into account multiple epitopes in an antigen. So let's say you have a lysozyme, which is very widely studied. There are multiple antibodies bound to different regions of these proteins. So all these epitopes were considered for a single protein in training and evaluation as well. It also uses hydrophilicity scale, Parker's hydrophilicity scale, amino acid occurrence that I talked about, then the number of contacts within 10 angstroms radius, and then the relative solvent accessibility. These are actually the features used by Discotope 1, and then Discotope 2 has actually improved, actually benchmarked these different parameters and used similar features but in a different way of calculation, and came up with a little bit better AUC of 0.73 compared to 0.71 of Discotope 1. So you can access Discotope, as I said, from the B-cell tools page. You can click on Discotope. The form is pretty simple. You provide a PDB ID. So that's a 4-letter ID. Or you can actually upload a PDB file, that's a coordinate file that I was mentioning earlier, and provide for which chains you would like to do the predictions for. That's why, again, I mentioned it's important to know there might be multiple chains in a complex, so you would actually want to know, okay, which is actually the one that you're interested in. And then you can select whether you would like to use Discotope 1.1 or 2, but 2 is the most recent version and which is recommended. So let's say you just take the same example just for an example to quickly go through the results, submit. You'll see a familiar interface again. This is the score threshold. The only thing that changes here is these two different buttons. So you have a table view and a 3D view. of the results. So all the positive predicted residues are highlighted in green above the threshold. Again, you'll see this information on which thresholds to use for different sensitivity and specificity that they correspond to on the help page or help tab. So the default is minus 3.7. That again corresponds to 75% specificity. So if you click on table view, you will get residue-wise score, and different parameters that are calculated by Epitope are also, and then the final score also, which is predicted by Epitope, is also shown here. And the epitope, the predicted epitope residues are highlighted in green. If you click on 3D view button here, you can actually go to JSmall visualization, and then these yellow residues here are the predicted epitope residues based on the threshold that you chose. And you can highlight individual residues by clicking here on CPK buttons and then visualize these. The demo was given by Nima yesterday. You can play around with this tool just by right click of the mouse. You can, let's say, calculate hydrogen bonds just by going to styles, hydrogen bonds, calculate, and you calculate hydrogen bond network in the protein, for an example. And of course, you can download these these images as well, as Nima showed yesterday. You can, of course, save all these results in a CSV format again, using this button here. And then coming to the second tool, which is ELIPRO for discontinuous epitope preparation. Do you have any questions on this tool? So ELIPRO is the second method that we have for predicting discontinuous epitopes. This method, basically what it does is approximates the protein's shape as an ellipsoid, and then gets protrusion indices for these residues here, and then cluster these residues based on how close they are in 3D space, and then predicts them as epitopes. So again, the input formats are pretty same. So you provide a PDB ID, or actually input or upload a PDB file. I would recommend you to keep the defaults unless you actually want to play around with these cutoffs. And if you want more information on these, I will be happy to provide you with that. But just to mention quickly, let's say if you are increasing this distance cutoff, you'll get more number of epitope residues, just for an example. So again, a modifying score will give you less or more number of epitope residues. So if you are getting too many epitope residues, you want to change them, you can play around with these parameters. But the defaults are recommended. So once you provide a PDB or input a PDB ID and submit the job, then you'll come to this page, which lets you select, okay, which chains would you like to predict the discontinuous epitopes for, chain A or chain B. If you select one of those, you'll get the result. You'll get to the results page, where you'll see your input sequence, the predicted linear epitopes, and then the predicted discontinuous epitopes. Again, you can visualize these epitopes in 3D using a JSmall applet by clicking these buttons here. Just to show you an example, if you click on that, it also tells you about the score and then the number of residues in this particular discontinuous epitope. If you scroll down, you can click here to view residue scores. You'll see a familiar interface again for the results. You'll see a chart, you'll see a threshold value, which here you cannot change because this has been fixed in the input parameters earlier. If you change those, you'll see that this is 0.5 score. If you change it to 0.6, you'll see less amount of epitopes. If you reduce the score, you'll see more epitopes. Same principle. Again, you have a data table, same format, you have the residue-wise, but the residue numbers here, they correspond to the residue numbers in the PDB file. And of course, you have a sequential numbers as well. With that, we'll do a quick exercise if you want to follow along. This is pretty quick. I've given a link to a dengue2 virus envelope glycoprotein. This is the PDB ID. You can click on it to go to the PDB website, get the sequences, and use Epipred and Epitope to predict B-cell epitopes. And then I'll go through this example, let's say after 10 minutes, whenever you're done. If you have questions in the meanwhile, please let me know. results. Should I wait a minute? Right. So even if you are done halfway, I'll walk through it slowly. Right. So you should, for Epitopes, it should be straightforward. You should have a result like that. This. But again, as you can see, there are just too many predicted epitopes here. So you can of course change this cutoff to 0.9 to get more specific results. Specifically, for this particular score threshold is 90%. So let's say you have narrowed down to these epitopes from Epitopes. Similarly, you should have, just by using default score threshold, you should have something, results like these for Discotope. What I want to focus on is if you are actually using a sequence-based and a structure-based method together, you should be a little bit more careful while analyzing the data later on. You can very easily see just by looking at the number here, this is 440, let's say 430 is the length, and then this is like 400 residues only for the Discotope. So the sequence-based and the structure-based methods give you a different length for the same protein. Why is that? So this is what I have done. So I actually took the sequence from the file that you must have downloaded from PDB and actually extracted the sequence from the PDB structure coordinate file, which is, I have labeled here as atom-seq. The Q is missing. And secres is what PDB calls its sequence, which it provides by default. You can quickly see here that the structure is missing residues. And these residues actually correspond to, and these residues are actually not resolved in the structure because of these loops might be flexible, disordered regions. In general, the resolution of the structure might also hinder to get a particular residue resolved. So if you want to actually combine these two results, get a consensus, you should actually do something like this. This is the simplest way you can analyze these results. Map the predicted residues here. So the Epitopes results are at the top. Discotope results are mapped in the bottom part of the alignment, which corresponds to the trees, like the sequence that I got from the PDB file. The predicted epitopes, I have mapped it using this yellow highlight from Epitopes and Discotope, say similarly throughout the sequence, and then the correctly. So I searched IEDB basically, got this assay which corresponds to the same dengue envelope glycoprotein. This is the PDB ID, and mapped those calculated epitope residues onto this with the green color, and you can see that they some of the predicted residues actually cover this region. But this is only one particular experimentally validated case. And this is actually one of the, as Bjorn also mentioned, that it's tricky to evaluate B-cell epitope prediction accuracies because of this issue, because of incomplete information in the data set that you might be using. So yesterday we saw Ibinome browser, and we were actually looking at this. Kerry was showing you the same protein sequence. If you narrow down this to the same part of the glycoprotein, envelope glycoprotein part of it, you can actually map more residues, epitope residues onto this alignment, and actually then see how the picture changes quickly. And this is one of the reasons why the evaluation of B-cell epitope prediction methods it's poor, because we might not have all this information in the data sets that people are using. And it's difficult to extract and combine this information anyways. But there is again one more downfall to even this, is that basically the whole surface of a protein can be target of some antibody out there. So people have actually developed methods which take into account antibody receptor sequence, the B-cell receptor sequence, and predict the epitopes only for that antibody sequence. So you have a specific prediction now, not a blind prediction where you can predict the whole surface accessible residues which might be targeted by some antibody out there. We don't know. But if you want something specific, you might actually end up using this algorithm. This is one of the first algorithms that was published. Bjorn was also part of this work. So if you're interested in something like this, I have provided the link in here so that you can use it. This is again the reason why I wanted to come back to the reliability of B-cell discontinuous epitope prediction tools. That the average AUC values they range from 0.6 to 0.7. And then again, these two are the protein-protein docking algorithms, AUC values, which do not fare well in comparison with the machine learning algorithms. In a little bit more recent work than that, from Discotope 2 paper, you'll see that Discotope 2 outperforms some of the older algorithms such as Epitopes, CAS, and SPR. But the performance is quite comparable with other methods such as Epitope and Epitope. Again, coming back to the issue of why the performance of the B-cell epitope prediction methods is poor, is the benchmark data set. The availability of data, because these methods, at least discontinuous epitope prediction methods, rely on structural information, which is in thousands compared to hundreds of thousands linear epitopes, let's say. And again, you might not have access to a complete biological unit or quaternary structure of a protein. And that's also an issue, and I guess one of you actually asked about the same question. Like if you have a protein complex with one or more units, can I actually predict epitopes for that? So Discotope and Elipro both can handle multiple chains. But unfortunately for Elipro, you have to go through the PDB file, change all the chain IDs to a single chain, like do AAA for column 22 in the PDB file, manually, or use some outside tools, and change the residue numbers, and then input it to Elipro. Discotope can actually handle multiple chain IDs. And again, there is a bias in the data set as well. Some of the proteins are well studied than the other protein antigens. So that's still there. Any questions on this continuous epitope prediction? If not, then we'll go for a 10-minute break.
Yes, 10-minute break, reconvene at 2:30.